Welcome students, welcome to the e-learning platform of Kalinga Institute of Social Sciences. Today we will study about the chapter 6 of unit 4 of biology paper. So, it is called Biotechnology, Principles and Processes. So, let me introduce what is biotechnology. Biotechnology, let me write it down. Biotechnology constitutes three words: bios, techno and logos. Bios means, as we know, bios means living or life. Techno means technical term, logos means to study. So, as a whole, biotechnology means it is the use of living organisms to study, with the study of living organisms to develop some industrial useful products for human welfare. आम ह्यूमन वेलफेयर हो पारे, एनवायरमेंटल वेलफेयर हो पारे, ए सब पे आमे किसी यूजफुल प्रोडक्ट तैयारी करिबा जो टेक्नोलॉजी द्वारा, दैट इज कॉल्ड बायोटेक्नोलॉजी. इट इज जस्ट यू कैन इमेजिन द मर्जर ऑफ information technology and biology. So, biology and information technology both are just merged here, just a technology and biology, those two fields are merged here to produce a field biotechnology. Let us start the history of biotechnology. Biotechnology, what is the history of biotechnology? Kebe biotechnology arambh hoithila. So, as we know, biotechnology can be of two types. As they have devised, it is one, traditional biotechnology, and second one, it is modern biotechnology. Traditional means we have been using the biotechnology tools in history, in our ancient life, in our culture, in our day-to-day life. Just for example, preparation of curd from milk. So, how curd is prepared from milk? Milk ru kemti khira ru kemti dahi tiyari hue. So, as you know, there is a bacteria called lactobacillus which is responsible for preparation of curd from milk. So, that bacteria which does this work, that means the involvement of an organism in some technology, that is the preparation of curd. So, that is the traditional biotechnology. So, traditionally we have been using biotechnology in our day-to-day life. There are several examples, but what is a modern biotechnology? Here we are using actual technical terms. We are extracting the genes, we are knowing the cause of the regions, what is the, how the products are produced, how the organisms they produce the products, we know the things, we are using genetics, knowledge of genetics, knowledge of molecular biology and knowledge of microbiology and mixed together to produce our useful, human useful or environmentally useful products in the field biotechnology. So, let me use biotechnology. What are the principles of biotechnology? To teach principles of biotechnology. So, principles of biotechnology, it consists of two parts again. If we consider about the principles of biotechnology, it also again has two parts. First one is genetic engineering, and second one is microbial growth. Genetic engineering, as you know, the engineering of genes. So, organisms they have genes which control all their activities. So, in genetic engineering we are just manipulating the genes for a particular action. Suppose amman naba insulin. Insulin ka na, what is the role of insulin? Insulin is a hormone which is useful to control the diabetes. Okay. So, insulin is produced by a specific gland in our pancreas, by beta cells in pancreas. They produce insulin. So, suppose some diabetic person they are lacking or they are unable to produce insulin. किसी डायबिटिक पर्सन जो माने इंसुलिन प्रोड्यूस करि पारु नाहंती. सो आम माने कोन करिबा ना देखिबा तांकर जो इंसुलिन प्रोड्यूसिंग जीन, जो जीन इंसुलिन प्रोड्यूस करछि इन द पैंक्रियास, से जीन को आनिकिरी आमे कट करिक आनिकी आम कि माइक्रो ऑर्गेनिज्म सपोज इन सम बैक्टीरिया धर ई कोलाई इज ए बैक्टीरिया व्हिच इज वेरी पॉपुलरली यूज्ड इन बायोटेक्नोलॉजी ई कोलाई एस्टेरिचिया कोलाई सपोज एस्टेरिचिया कोलाई रो जीन भितर आम इंसुलिन जीन ह्यूमन इंसुलिन जीन को इंट्रोड्यूस करदेबा okay that introduction part that is a merger of genes or the recombination of genes a mammalian gene insulin gene which is our useful or desired gene that is merged with the gene of a bacteria prokaryotic gene ये जो मर्जर कर दिया गला दैट इज कॉल्ड द इंजीनियर दैट इज अ टेक्निकल वर्ड द टेक्निकल प्रोसेस इज कैरिड आउट दैट इज कॉल्ड जेनेटिक इंजीनियरिंग देन तापर हम कौन करिबा ना जो बैक्टीरिया टाको आम तैयारी कलो दैट इज ए रिकम्बिनेंट बैक्टीरिया जेनेटिकली मॉडिफाइड ऑर्गेनिज्म दैट इज कॉल्ड जेनेटिकली मॉडिफाइड ऑर्गेनिज्म आम ताको जेनेटिकली मॉडिफाइड करिचु दैट बैक्टीरिया को आम से बैक्टीरिया को माइक्रोबियल ग्रोथ रे हम तो ग्रोथ करेबा माइक्रोबियल ग्रोथ कौन दैट द ग्रोथ ऑफ माइक्रो ऑर्गेनिज्म सपोज गोटे बैक्टीरिया रो कल्चर ऑफ बैक्टीरिया माने कल्चर कलो आम तो कल्चर मीडियम रे छाड़ी देलु सो कल्चर मीडियम दे आर गेटिंग मच मोर सफिशिएंट न्यूट्रिएंट एंड दे आर growing rapidly in an exponential way just like 2 to the power n in this fraction so ये जो microbial growth बस ये जो बैक्टीरिया माने ग्रो कले टू प्रोड्यूस ए प्लेंटी नंबर ऑफ बैक्टीरिया से बैक्टीरिया मानव पाखरे इंसुलिन जीन रही जाइछि जोटा को हम जेनेटिक इंजीनियरिंग तैयारी करिछु से बैक्टीरिया माने वर्तमान इंसुलिन प्रोड्यूस करिवे नाउ दे विल प्रोड्यूस इंसुलिन सो इन प्रिंसिपल्स ऑफ बायोटेक्नोलॉजी we follow microbial growth of the microorganism से बैक्टीरिया को ग्रो करेबा ता कोटे कल्चर मीडियम दबा से ग्रो कला plenty number of bacteria are now produced now these bacteria having the insulin gene they will generate the insulin gene product that is insulin is the protein which is the product of the gene insulin gene so बर्तमान में इंसुलिन पाई गले माइक्रोबियल ग्रोथ रु सो से कल्चर मीडियम रु हमें नाउ वी कलेक्ट एक्सट्रैक्ट द इंसुलिन the protein insulin protein and will supply it to the disease person जो मान डायबिटीज तांको बर्तमान इंसुलिन हमें सप्लाई करि परिबा सो एटा होची प्रिंसिपल्स ऑफ बायोटेक्नोलॉजी दीटा कम तुमनो देखै देली नाउ व्हाट आर द कंपोनेंट्स इन बायोटेक्नोलॉजी रे को कंपोनेंट्स सब इन्वॉल्व हुए एबे हमें से विषय देखिबा को कंपोनेंट्स सो यूज हुए सो हमरो जेनेटिक इंजीनियरिंग हम आरंभ करबा फर्स्ट पार्ट द जेनेटिक इंजीनियरिंग इज आल्सो नोन एज recombinant DNA technology. Let me write it down. Recombinant DNA technology. It is shortly known as RDT. Recombinant DNA technology, shortly known as RDT or rDNA technology. So, what are the components of rDNA technology? rDNA technology बा recombinant DNA technology रे कौन सब पार्ट अच्छी से सब पार्ट को हमें देखिबा सो कौन पार्ट अच्छी ना तो चारिटा मुख्य मेन जिनस दरकार 4 parts are here four tools are here so what are the tools required for recombinant DNA technology the first one the enzymes what are the enzymes these are 4 types of enzymes which are required for recombinant DNA technology then there is a cloning vector will come into it later so first one the restriction enzymes have 4 types of enzymes which are required first one restriction endonuclease in short it is known as RE second one DNA ligase, third one alkaline phosphatase enzyme and the fourth one DNA polymerase enzyme. And after the enzyme there are also required a cloning vector which can transfer our desired DNA into the host organism. Then host organism where our desired DNA will multiply just like the bacteria E. coli will cultivate the host organism to produce the desired gene product. Desired foreign DNA. Desired foreign DNA is our required foreign DNA which is our target DNA. आमे माने जो target DNA जार product आमे चाहूँ से product टा जो थ्रू बाहरी बो that is called desired foreign DNA. So let me come to it step by step. Enzyme by enzyme we will discuss the enzymes. Okay. So एबे आमे देखिबा restriction endonuclease. Restriction endonuclease कौन आउ कौन यार दरकार So restriction endonuclease are a particular type of enzymes which comes under a group of enzyme, a whole group of general group of enzyme that is called nuclease. So nuclease are a group of enzyme. Their work is to digest the DNA or nucleic acid. So they are of two types, exonuclease and endonuclease. As the name shows, exonuclease digests the DNA molecule from two ends, either from 5' end or 3' end. As you know, DNA has two ends, one is 5' and 3'. So DNA has two ends, 5' and 3'. So exonuclease can digest the DNA from either 5' end or 3' end, any end. Endonuclease can call the DNA from between these two ends, any point between these two ends, 5' or 3'. So that's why they are named so. So in our consideration, there is endonuclease called restriction endonuclease. So restriction endonuclease जोटा कि देखा जाए स्पेसिफिकली आम ताको यूज करूँ बायोटेक्नोलॉजी रे, दिस आर जनरली एक्सट्रैक्टेड फ्रॉम द बैक्टेरियाज आम मैंने विषय नेक्स्ट सेशन रे पढ़िबा रेस्ट्रिक्शन एंडोनुक्लेज विषय। थैंक यू।